for today, again a twofer, two from our Department of Anthropology. We've called often upon people from our Department of Anthropology. I know many of you were here in the fall to hear Tawny Tidwell, the only Westerner credentialed in Tibetan medicine talk. Just finished her PhD here. We've had Mel Connor, we've had Brad Shore, we've had Aaron Stutz and his wife Liv. And today, thanks to a wonderful article I read in eScience Comments, we have a combo of Assistant Professor Jessica Thompson and PhD candidate Kendra Sirak. And just a tiny bit of information about each. I don't want to take too much of their talking time. So first of all, Jessica came out, well, I don't know where you actually grew up, but her BS was University of New Mexico in anthropology with a minor. Yes, Mary Lynn, yes. Right, she's making hand gestures, okay. University of New Mexico. And actually, I wonder if you might have overlapped there. You'll have to check. Mary Lynn went back later, right, to get your... Retired. Yeah. Anyway, so that was a BS in anthropology with a minor in biology. She then headed to England and got an MPhil at Cambridge. Then she came back to the Southwest and got a PhD at Arizona State. And then she spent 5 years as a postdoc in Australia before coming finally to Emory in 2015. All those years, though, all over the world, but I think her heart and much of her work was always in Africa, and she'll be talking about her work in Africa today, and particularly in Malawi. And she is the principal investigator and director of MEMSAP, which is Malawi Earlier Middle Stone Age Project. Kendra came out of Northwestern, right? Undergraduate degree in anthropology, came to Emory and has finished her MA here at Emory, and she was working with George Armelivus, and many of you, I know, knew George. When George passed away, his work did not stop, and Kendra continued working with, among others, Jessica, and has been to Malawi and done a lot of, the two of them working together on site, in the field, and then also analyzing the results of what's been found. Kendra is particularly interested in the intersection between genetics and anthropology, archaeology, and is particularly interested in earlier Nubian populations. But what they're going to talk about today has to do with a period of time in Malawi, in that whole region of Africa, when old ways of life, the hunter-gathering life ways, were giving way to the big change, right, brought about by herders and farmers. So let me turn it over to them. They're going to take turns and allow plenty of time for questions in the end. Thank you.
That was that's loud. That was an amazing introduction. Thank you. And I was really impressed because I'm chairing the colloquium committee in anthropology, and I can never remember everyone's details that way without a was there for 3 years, and I guess I grew up in Eastern Washington. So Washington State University was where my parents worked, and I've always been kind of around academic type people. And when I was growing up, I just felt like, okay, well, that's what you do. You grow up and you become a professor. That's normal. And yeah, it's been an interesting reality to learn otherwise. But also your introduction with respect to Kendra and George was wonderful. I never actually met George. I believe when I interviewed here, he was still here, but it was only a couple of months later that he passed away. So I never did have a chance to meet him, but I obviously very familiar with his work, and was very fortunate that Kendra was able to, I feel kind of strange saying that I'm mentoring her because she does a lot herself. She's very self-mentoring. And it was mostly because of my experience working with bones that I kind of fell into that position, and also because I have a long-standing interest in ancient human DNA, ancient DNA in general. But what I have been unable to kind of do with it is because I usually work in really old time periods. So you mentioned the Malawi earlier Middle Stone Age project. That was my first project in Malawi, and I still have that on my website because I still have the website up, and I have not had a chance to to put the new project up, which is the Malawi Ancient Lifeways and Peoples project. And I needed a new title because the time period was wrong. It's not the earlier or the Middle Stone Age that we're actually studying. And kind of an interesting thing is that even that project wasn't quite what we turned it out to be. We thought it was going to be one time period, it ended up being another. So I've been learning a lot about being flexible. Anyway, here is what we'll talk about. If you've been seeing any of this in the news, this is Horror Mountain, and this is the kind of publicity photo that I took quite accidentally. We were off, if you can imagine, there's all of these big kind of inselbergs that stick up out of the plain, and there'd be one here, one here, one here, and we were up here on this one looking for some rock art that we had heard about, but that we hadn't confirmed actually existed. So we were poking around up there looking for it. I turned around and I just saw this, and the sun was setting, and I took out the camera, and I think I even forgot to put it on the high resolution, and I just took a quick photo. Turns out to be one of the best photos I've taken out there. But Horror Mountain is here, and just to kind of orient you, and I will zoom out and we'll look at the broader picture. You're probably like, would like to have a map of Malawi or something. That will come. You'll see a lot of maps of Malawi. But this is what it looked like from the side. So these are two little inselbergs that are actually over here, if you can imagine. So we're standing here, and we're looking at the shelter. The shelter is just this little thing here, and you'll see more pictures of it soon. So there we go. It was actually excavated in 1950, and it had a single report that was written about it, and then it would kind of show up in other pieces of literature, but it was completely undated. And what the original excavator had found was a rich archaeological and fossil record, including 2 human skeletons. So human skeletons from hunter-gatherers that we suspect are quite ancient, they're rare. Africa is a big place, so even if you start looking at all of the skeletons and start calculating them and trying to find out where they are and sort of hunting them down, which is what I've been preoccupied with for the last couple of years, you find that there's still only, maybe 50 or 60 of them. And it's very rare to find 2 complete skeletons like this as well. The South African record is a little bit more robust. So when I say that 50 or 60, I'm talking about kind of the Central African belt. And then in North Africa, there are a lot more as well. So it kind of depends where you're working. Sometimes you have a lot of opportunity to study the ancient human remains, and sometimes you have almost no opportunity. And Central Africa is one of those very little opportunity kinds of places. However, we did get some publicity because we did manage to extract DNA from those 2 skeletons. And Kendra did not do the sampling on those 2 individuals. No, you did. You processed them. Yeah, but you didn't go physically acquire the bones like she did for the other remains that we had in this report. So she's going to go through what she did. It turns out that when we published this, there were 15 specimens that were published. It's the oldest ancient DNA in Africa, and up to the point of our publication last year, there was only 1 that had been published. So 15 was a substantial increase in the number of ancient genomes that we have accessible to us. And in addition to that, that helped us date the site because we were able to get a direct age on one of those skeletons. And now we know that they're at least 8,000 years old. What I'm going to do is I'm going to walk you through kind of that process of discovery. And first, okay, there's my project. This is a kind of a stock photo. You'll actually be able to find this photo on the internet of a Khoisan hunter-gatherer with his bow. And then my brother he's a graphic artist, and he's kind of modified it to make it look very animated. And then this is the shape of Malawi here. So J. Desmond Clark is some people call him the grandfather or the father of Africanist archaeology. He was living in Africa for about 30 years, and he was born in 1916. He died actually in 2002, and here's another person that I never had a chance to meet. The year I started my PhD. So I was not able to actually ever meet him in person, but he's my academic grandfather. So he was the mentor of my mentor. And I find it amazing because when I was doing some research just for the biography part of it, he died on Valentine's Day, and my grandfather died on Valentine's Day. So, you know, I mean, it's all of these funny things kind of pop up. And what we're going to do is we're going to go through, you know, the archaeology, obviously, but the process of trying to reconstruct also what other archaeologists have done is in and of itself kind of an archaeological process. So I'll hopefully be able to take you through that. So we'll know at the end, hopefully, a little bit more about what actually happened in the past. But also, you're going to see a lot here about how standards and approaches have changed and how that really affects what it is that we think we can say about the past and the confidence with which we can say it. There are also issues of cultural sensitivity that I think we're a lot better at dealing with now than people were in 1950 when he first excavated at Hora. So we'll touch on some of those issues as well. And then the utility of going back to old sites. I knew that was going to get me the thing going the wrong way. Right, so here's Clark with Louis Leakey in 1950. So this is when he was a young man. He just finished his PhD. He was living in Zambia. And you can see that he has this kind of archetypical, you know, Indiana Jones sort of archaeology style about him. He also had a way with words. He was very prosaic. And he starts one of his papers. This is the paper where he describes the Hora site. And Nyasaland, of course, that's what Malawi was Nyasaland when it was under British colonial rule. And it says, Nyasaland has long been the Cinderella of Central African prehistory. So I suppose what he means by that is something that's really important, but overlooked most of the time. And I've certainly found that to be the case. So that's a direct quote from him. Here's Cinderella in Malawi. And there she is, right there. So with the fedora. You can see that just kind of immediately there's some things you can tell about it. Its position is not where you usually see people working when they are going to give a presentation about, say, human origins. They'll be pointing to places up here, or they'll be pointing to places down here. And that's how I got into the area, was I had done my PhD work down here, and then I had done some like fun work up here. And I realized that there was an enormous gap in what we knew about, well, this, right? So I went to Malawi and I started a project there, and that was on much, much earlier time periods, probably about 100,000 to 60,000 years ago. But it got me into the region, and I started to learn more about it. And I could see that there was a lot more potential that it had in other time periods. And more to the point, with respect to my personal interest, I kind of felt like maybe if we knew more about the recent past, we'd be able to extrapolate a little bit. We'd be able to push that back and use that as a window into the even deeper past, rather than taking such a giant leap. That was the hope, anyway. All right, so here we are. Hora is here, and this is another site that I'll talk about, which is Mazinga. So I've zoomed in. This is the shape of Malawi, and you can see where it's positioned relative to the country. The capital is here, so when we go there, we fly in there, and then it takes us about 4 hours to drive up there. Maybe more like 6 hours, because, you know, I mean, you look on Google Maps and it doesn't calculate the fact that the road is maybe not there from year to year. So, yeah, it usually takes us about 6 hours to get to our field site. But you can see there's a significant topographic gradient over even a pretty short distance. So here's the Kasitu Valley. That's a river. It's not a massive river, but it is a major drainage in the area. And up here is a plateau, and it may not look that substantial, but it actually goes all the way up to about 1750 meters. And then up here to the north, so you can see this dark spot here, that's another plateau that reaches 2,500 meters. It's very, very cold up there. And I'm pointing that out now because some of the ancient DNA that we got was from a cave up there. So we have been kind of roaming around in this area. And the Kasitu sites themselves are still pretty fair distance up at about 1,400 meters as compared to Lake Malawi, which is only at about 500 meters. So, you know, when you're driving it or walking it, it's a pretty significant climb. You definitely feel you're climbing. This is what the shelter looks like. It's Cinderella, a little bit nondescript. It's just basically a boulder that's sticking up out of the ground, and it's tumbled down the side of the mountain. We saw all these Inselbergs sticking up out of the plain, and we got very excited, and we thought, wow, there's going to be so much archaeology. And there is a lot of archaeology, but there are not a lot of really substantive rock shelters to contain that archaeology. It's more kind of diffuse across the whole landscape. And then you get very lucky when you actually find one of these shelters. And the reason for that is because the sides of the Inselbergs are so steep, and they're made out of this kind of schist granite, and it kind of exfoliates out like this. So it doesn't really create an opportunity for big cracks and overhangs and things like that to form. And it's a very active weathering environment as well. So the cracks that do form, they fill and they collapse pretty quickly. So to find something like this, and to know that it has some antiquity to it, is a pretty rare opportunity, actually. And I was surprised by that. I thought there would be a lot more places like this. So you can see when we went back in 2016, here's what it looked like. This was an original archive photo, the black and white one from 1950, when Clark was working there. So you can see the first thing you would do as an archaeologist in 1950 is just hire a big labor force and have them clear out everything. So we don't do that now. We clear what we need to clear, and we try to be as non-disruptive as we possibly can, given the fact that by nature our work is kind of destructive because we are digging things down, and once that stuff is out of the ground, you can never go back. You have to do the best job that you can at the time that you're excavating it. And I think that in the 1950s, there was a sense of that, but I think there was also a sense that there would always be more, and that it was kind of not an infinite, not a finite resource. And now I think, at least in archaeology, we're very aware of how rare these kinds of places actually are and how important it is to conserve them. So the more we can do small samples non-invasively, both in terms of how big our excavations are, get the same amount of information that you could have got from a huge dig back in the day, then that's going to be the preferred option. So, okay, I have some information about them there. You can see, when it was excavated. This is another site on the mountain, and you can see the date range there. And this kind of drew my eye because although I was working 100 to 60,000 years ago, this is still kind of old. That's not really recent. And I thought, wow, maybe there's something else even older. And I wanted to be the person who would go in there and find that, especially knowing that sites like Hora 1, which is this one, had never been dated before. So it was kind of a tantalizing place to start. Now, this was Clark's excavation from the 1950s. You can see they dig straight down. They dug in feet, right? Not meters, which we do meters now. So everything we've had to do when we go back to old photographs or drawings is you always look at it and then your scale is completely wrong and then you have to do a bunch of converting. And it's not like it's a hard conversion, but just mentally it's a bit challenging when you're used to looking at a photo to try to figure out what your scale is. And there's always this big question of how old it was. So you can see, here's what it looks like now. Somebody has filled the pit, and I will say I've been chasing Clark's excavations around Malawi since 2009, and this is the first one I've seen that they've actually filled back in. So that should be standard practice. If you don't fill it back in, it just does this. It just slumps, and then all that carefully stratified material that you've been so careful to excavate one by one by one, it just pours into the hole and gets completely jumbled up, and then you have an inability to really date it or know what's associated with what. That's the most important thing in archaeology, is to know what belongs with what and how old is it. So nowadays we will sandbag it and then fill it, and of course you don't want the sandbags to stick out, because if they stick out, people come along, they may be not familiar with what's going on. They say, oh, somebody's got some treasure here, right? And then they pull out the sandbags, and then they do the same thing, which is why community consultation has got to be one of the key elements to any project. You have to get into the community, and you have to talk to people who have the authority to disseminate information to everybody so that they know that whatever's going on up there in that shelter, the chiefs have authority over that area. They have respect for the chiefs and for the local authorities and the traditional owners. And so if you can talk to them and work with them, then it just makes everybody's life easier. And you find that they're usually very enthusiastic to be a part of it as well. They like to feel like their little area is of international significance. I think we all know what that feels like. All right, so in Clark's words, I'm going to use Clark's words to describe this. He's got 3 main layers. I say 4, but that just includes this surface, superficial stuff. So under the superficial deposit, and I can just imagine, because he was British, I can imagine his voice, occurred one of blackish earth containing an industry of Gachikufu 3 affinities. Now what that is what you would do back in the day is you'd find a site, you'd excavate the whole thing down, usually huge excavation. You collect together all of the things that you think look meaningful. So if you've got some pottery, or if you've got some stone artifacts and you think maybe they're of a shape that is specific to that site, and that people intentionally made them into that shape, then you might name that an industry that you name after that site. So this industry that he's talking about is from Nashikufu Cave, which is actually 300 kilometers away in Zambia. But that's the best he had to work with in 1950, and that's how spotty still a lot of our data points are, where the only reference point that he had was that far away. So he saw something that looked a little bit similar to that, and he named it Nashikufu. So this is what he, and this is my favorite, the degenerate quartz industry. I have a colleague who says she's going to name her metal synth band the Degenerate Quartz Industry. That's going to be her metal band name. But it is kind of all of the raw materials that you would use in terms of stone. So you're going around the landscape, you're looking for rocks that you can break to make into stone artifacts. There are nice rocks and there are not nice rocks. And this area is full of not nice rocks. It really is kind of frustrating. And it's not necessarily because it's a degenerate industry. It's because there is not much available that you can work with that's going to break in a way that's that productive. So it tends to be really small chunks. It's kind of blocky, so when you hit it, it just sort of shatters. You don't have nice, beautiful flakes like I'm sure you've seen other people presenting this before. So Nachikufu degenerate industry probably has a lot more to do with availability of stuff on the landscape. Now, Clark reported that he had found some beads, 3-shell beads, and the beads were of this kind of giant land snail that lives in Africa. And I'm holding my hand out like this because the snail would cover your whole hand. It's a huge snail. And there are a lot of these snail shells in the deposit, and people were making them into beads. And you find this all across Central Africa and a little bit into Southern Africa. There's almost no research on it. Right now, I know of a paper that's in review that is the first ever paper that's explicitly on snail shell beads. It's the strangest thing that nobody has ever paid attention, and that's because of this kind of cultural industry approach to things. So you come to a site and you say, all right, there are shell beads, tick, okay, we've got small stone tools, tick, we've got some pottery, it looks like this, tick, and then you call it an industry, and it just becomes part of the characteristics of the industry. And then you don't really go into the details of how the beads were made or how they were maybe traded, and a really important part of the cultural system is there. That's one of the things that we're going to focus on in our future work. You'll also notice that we recovered 41 beads, and we excavated 1/4 of the deposit that Clark excavated. So we'll be looking at a substantial change in excavation methodology. What that is got to be sieve size. So he either wasn't sieving at all, he wasn't putting all of the dirt through a sieve, or he was using a very big sieve size, so that all the tiny shell beads were just falling through. And then that ends up in a big pile on the side of the site, and then you kind of pick through it, and you find something that looks like a big bone, you know, something that looks really important, and then you pull that out, and that gets in your box, and that's what ends up in the museum. Whereas these days, we recover everything. And if you want to, you're very welcome to come to my lab where I have all of this, and you can see the stuff that we're dealing with. And it takes armies of undergraduates to help me work through it, because we've got to sort it and clean it and label it and process it. And it's just, even if it's not going to ultimately tell us anything, we don't know if it might tell somebody in the future something. So as custodians not only of archaeology in the past, we have to be custodians of archaeologists in the future and think about those unknown analyses that we can't anticipate and do the best job that we can to try to get the collection in a place where it can still be useful to people in the future who might have things they can do with it that we can't even imagine. And even now, the fact that we do sieve, okay, yeah, we use a 1 millimeter sieve, but we're still losing the dirt, right? And now I wonder, the dirt, where are we supposed to be keeping the dirt? And even last year, there was a paper that was published where they got ancient DNA out of dirt, and they found Neanderthal DNA in dirt. And I thought, oh. But you know, what we do is we take a sample of all of the sediment every single time we excavate a new little area. So hopefully there's at least something there for the future. He found 2 burials. I mentioned these burials before, and you can see they're quite complete. This is a female burial that he recovered, all tucked up in there. And they're not buried with anything. There's no pottery, there's no stone artifact. I mean, it's all in the sediment around them, but it's not clear that these were objects that were placed into a grave, specifically like as grave goods. And often in the later Stone Age, which is this time period, you'll find that people will be decorated with beads and things when they're buried, and these people apparently were not. At least Clark didn't report it, and you can't see it. And this is the only surviving photograph of that burial. This is really counterintuitive. It's terrible. He was telling me that the buttons go opposite ways, and I was like, yeah, I got that, but no, very counterintuitive. So there's a male and a female, and he's calling these within the Nachikufu 2, which is just basically the same as this, but without pottery. And that way. There's your degenerate quartz industry. That's what it looks like on the surface. I don't have any photographs of the material we have in the lab because we're still processing it. And I just realized that when I was putting together the talk. But we have many, many kilograms of quartz. If you want to come look at it, you certainly can. It's being processed and analyzed. And here's how he describes it: small, straight microliths, so very tiny little toned jewels. Bipolar cores just means a little lump of rock that you've smashed and it shatters. And then these are just kind of things that people used to describe back in the day as tools. But in reality, probably most of these things were used as tools by people. You break the rock, you pick whatever one looks like it's going to do the job, and you do that. You don't. I think archaeologists have always had this tendency to find things that are of a specific shape and then try to say, okay, well, this person certainly meant to make it into the shape, and there's a cultural difference between this group and that group. And that is true to some extent, but for the most part, we're finding that most of this is technologically driven, not driven by the kind of cultural identities of people, but through cultural learning that is constrained by the available materials you have to work with in the first instance, and secondarily by just the technological know-how that you have to be able to manufacture things. And at the end of the day, with these little lumps of quartz, there's only a few ways you can really break them. So not a lot of it is culturally able to be shaped by cultural decisions because they're just so constrained by the properties of the materials themselves. Much less the case with things like shell beads. Those you can actually manufacture and do a lot of different things with, so that you might be able to discern different cultural learned ways of doing things between cultural groups. So if we go to one site and we find they're using a lot of red pigment to color their beads, or this site over here we find that they're drilling it before they shape it, and this site maybe they're shaping it before they drill it. Those are culturally learned differences that tend to be, the child is learning and they say, okay, and they watch what's going on and they learn it that way. And it's not necessarily because it's a better way, but because it's the cultural pathway. So the beads have become very interesting to us for these reasons, because we think they might be a better avenue than the stone artifacts into what was actually going on in terms of cultural groups. Now, how old is it? Here's what Clark had decided. He did not know how old any of it was. 1950 was the year that radiocarbon dating became a thing. So he didn't submit anything for radiocarbon dating. He just went on and did the rest of his work in Zambia. He came back later, a couple of decades, to Malawi again, and they never dated the site. In Zambia, they dated the type site of the Nachikufun and found that the stuff associated with one seems to be about 18,000 to 10,000 years old. The stuff associated with 2 goes big range, 3,000 to 10,000. And then the stuff associated with 3 is the youngest, as Clark predicted. So we had this before we went in. And we thought, okay, well, maybe it's about this old, right? And the burials would be, therefore, maybe between 10 and 3,000 years old. This was the inference. Reviewers were not interested in this interpretation. They wanted dates. They wanted us to go to the site, excavate the site, date the site, and then ask for money to go to the site, excavate the site, and date the site, once we knew how old it was. So we did that. And I have many entities to thank for that. But I think we're in a good position now because we actually do know how old it is, and this is how old it is. So this is just the top meter. Remember, Clark excavated 2 meters, 2 plus. So there'll be another one like this that we're going to find this summer when we go back. And down here at the bottom is 14,000 years old. And right near the top, we're getting 9,000, which is weird because you would expect there to be, recent stuff, 1,000, 3,000, and 4,000, and we're sort of missing all of that. So there's pottery up here. There's evidence of ironworking. It's been kind of mixed into this top part, this top 20 centimeters. There's a lot of termite activity at the site. You can see the termites are building big mounds against the wall. So there's concern that they're moving things around in such a way that you won't be able to discern any stratigraphy. We can see stratigraphy. You can see there's this kind of homogenized area here. This is the termites. They have homogenized it. And in spite of that, we got some dates here, 8,500, 8,900, 9,200. So they're all in stratigraphic sequence in spite of that homogenization. So it seems like it's a fairly local kind of just mixing and churning that's going on, and that the basic stratigraphy has been preserved. And then away from the wall, we have real stratigraphy. So stuff that is definitely still intact. And these layers that you're seeing here are layers of ash. And right down here is a huge lump of ash. Enormous. It's 2 meters by 1 meter thick, like this. Just huge lump of ash with almost no charcoal in it. So we're taking some micro samples and we're going to try to look at them in thin section and figure out what kind of plants they were probably burning there, because if there's no charcoal, it probably wasn't a wood fire. It was probably a big grass fire or something like that. Maybe we can even see the way that the fibers have been arranged, and it might even be revealed to be some matting or something like that. So this is very exciting to see, the possibilities there. And below that, we have some ostrich eggshell fragments. That's what OES is. And at the bottom, about 14,000 years old. So I don't know how old it's going to be, but this is a very nice stratigraphic sequence. The dates are all in order. So I'm very excited about what we're going to find when we go back. And I just want to point out those ostriches, they're actually pretty important. So we'll get back to those guys later. And as Clark, ever the kind of future-looking man that he was, noted that they're very interesting because there are no ostriches that live there today. And I thought, well, maybe that's because human disturbance, they've been hunted, I don't know. But no, there's actually a biogeographic zone right there where the ostriches just don't like to live there. It's not the right environment for them. So there's ostriches that live way to the north and way to the south, and they do not live in the area where we live now. So either these ostriches are indicating a serious change in environment in the past, or they are traded, and they've actually become trade goods. And so we'll get into the ostriches a bit later. Right now, I want to point out what we found here. I don't know if you can see my complicated legend says human remains, right? So we actually found another skeleton while we were excavating over the summer. And it was just, this is a distal humerus. Come on. This is a distal humerus, right? So it's the elbow joint. It was right there where we're very excited. Yeah, and so excited. And this is what it looks like. You can see it actually in situ, so it's sticking out right there. This is it kind of sideways, so you can see that shape is the same as this shape, but this is like upside down. And then this is that huge lump of ash I was talking about. This whole thing is ash with little micro layers in it. And this is more human remains here. So here's what it looks like in plan. So looking at the top, there's a big feature of very clearly burnt material. And remember that giant lump of ash had almost no charcoal in it, and it also had almost no bone and snail shell and stuff in it, like the rest of the deposit. So it looks like it was just a one-off event, huge fire. We must have been talking about an enormous fire, and to leave that quantity of ash, and almost none of it being wood. And then below it is this pile of human remains. So we tentatively called it a cremation, but we need to confirm that by actually comparing it to known cremations and seeing if it was accidentally burnt just because it was in the wrong place at the wrong time, or if it looks like it was intentionally separately burned and then maybe became incorporated into this larger burning event. We're also very interested in knowing its relationship to the burning event. And so we dated the ash on top, 9,400, and then we dated a little tiny piece of charcoal right below the human remains, and it's exactly the same age. So this is all one thing that happened here. Someone died, and someone was involved in the giant burning event, and there's no teeth, and there's no skull, and there's no vertebra, and there's no ribs. There's only long bones there. So it looks like maybe somebody had been maybe cremated, and then the long bones pulled out and then reinterred there, or possibly left exposed, and then the long bones were pulled out as the remains and then burnt as part of this feature. But it's a very interesting feature regardless. And then you can even see here, once we reconstructed a little bit of it in the lab, this is a femur. So this is a human femur, and we got most of it. And both of them are there. So there's two femora, they're matching from either side. You can see they're not in great shape because they've been heavily burned. That's not great for ancient DNA prospects because burning is not helpful. But what we did do is we took this little tiny piece here, and we'll see, because we were careful to pick one that was very dense and that was away from any of the really diagnostic parts of the bone, so that hopefully any future bioarchaeologists will not be bothered by the little piece that we've taken out for that. At the very least, we can confirm that it's not present. And the reason, of course, we did that is because there's two other skeletons with ancient human DNA in the site that are 1,000 years younger. So it would be wonderful to be able to see if there is population continuity at the same site, in the same place. Certainly, they're interred in a very different way. right? So this was a cremation, and then the others are actual burials. Right, so it's time finally to zoom out and see where we are. Just to put it in context, where there are other sites with human remains, you'll see the blue. That's all sites that have human remains, and they're usually like one tooth or a few fragments of something. It's not complete skeletons in almost every case. There's one exception, and that's up here. There is most of a skeleton, and then about 12 other individuals. And down here, there is most of a skeleton, an infant, and then there's some other fragments. But for the most part, it's just fragmentary pieces that kind of show up in these sites. And one of the big questions is this part of like the cultural behavior of people to kind of carry pieces of their ancestors around with them, to engage in different kinds of ritual activities, or is it actually just because people are passing away, they're getting buried, and then carnivores are coming and disturbing the site and pulling them away? And so maybe they're just showing up as fragments because of an accident of natural processes. And we'd like to be able to start to look at some of the other sites too, so we can actually get a comparative view. Rock art is abundant. You can see this red stuff, and the rock art is almost certainly made by hunter-gatherers in the area, probably pretty recently though. The reason being because these materials, the rocks exfoliate so quickly that it's unlikely that they would preserve rock art longer than a few thousand years, which is kind of unfortunate. And then, of course, this is Hora. This is another site where we got ancient DNA from 3 individuals and one where we got ancient DNA from 2 individuals. So Kendra will talk about those. Right, that line is because the rock art down here appears to be stylistically different from the rock art up here. Up here, it's all things like abstract patterns, nothing that looks like an actual animal. And then down here, you get both. You get the abstract patterns, and you also get the more naturalistic kinds of things. So it's possible that you have some cultural differences between those 2 areas. Zooming way out, here is the regional significance. You can see where it is positioned relative to the living hunter-gatherer that we could go potentially and study today, or at least to try to understand something about how hunter-gatherers live and interact with their environments. You might go spend some time with them, and they're all in these environments here, like the deep African rainforest here. This is extremely arid part of Tanzania, and then down here also very arid. There's a reason for that, and that's not because people have a preference when they're hunting and gathering to live in really severe environments, right? It's because every other place on Africa, the African continent, is occupied by people who produce food. So agriculture and pastoralism. And where those things are not possible, hunter-gatherers have persisted. So we have an extremely biased sample of trying to understand how it is that hunter-gatherers actually lived, their day-to-day activities, and how they organize themselves socially. We can get some clues from living people, but we really don't have any idea how they were living in these environments across here, which is a very distinctive kind of environment. You can see this is our study area there. It's in the blue. So these are the phytogeographic zones of Africa. And you can see that modern hunter-gatherers live in very specific marginal kinds of environments that are not good for food producers. And we're in an area that's quite good for food production. And it's in this Zambesian phytogeographic zone. So we don't have any access into that unless we go to the archaeological record, because we can't go and observe people living there now. And there isn't even any surviving oral history. There are some hints of it in oral history. There are hints of small people who used to kind of live in the bush and they would hunt. But it's really not going to give you much information beyond that. And it's difficult to confirm, when the observations were made. And of course, they will have been made by food producers as well. So they're going to be biased in what they're observing. And so I would argue that the archaeological record becomes imperative if we want to actually understand these environments, which are really the environments that dominated many of our own species' early migrations around Africa. So as Homo sapiens was emerging as a species, of course, we were in lots of different parts of Africa, occupying lots of different niches. And we can only directly observe that behavior in just a few very specific kinds of environments. So my argument that I'm trying to make is that we have to go to the archaeology if we're going to try to understand human interactions with each other, with their environments, in this very large area. And we know from the archaeological record that people have been there for a really long time, at least 230,000 years. So certainly it's an important area. That's what it looks like, just to give you a sense of what the Zambesian phytogeographic zone actually looks like when you're standing in it. This is taken from Hoare Mountain, and you would imagine it might look something like that in the past, right? You wouldn't have all this cleared land for agriculture, but you would see smoke rising up in the distance, and you would see the mountains kind of hazy in the distance. And especially during arid periods, you would have a very hazy kind of outlook because there would be a lot of large herds of wild ungulates moving through this valley, and they'd be kicking up dust, and they'd be making a lot of noise. There aren't any there now because so many people, it's a very densely populated country, so there's so many people there with their little farms. But in the past, this would have been a big corridor for large ungulate herds to be living. And there's some up there. These guys survive because they're up in these high montane grasslands. So remember I mentioned that other site that's way up on the plateau? This is what the environment looks like more up there. And this is the lake itself. So you're kind of going from lake through this up into this. And that's the diversity you can get over just 100 kilometers of horizontal distance. It's a very diverse kind of landscape over short distances. In the valley itself, I started to come up with some ideas about how it is that we can try to approach. Okay, I could say, let's understand what they did, but that's a very vague thing to say, right? So what is it exactly that I want to know about these ancient lifeways? And one of them is, how much did fluctuations in the environment in the past actually impact hunter-gatherer social organization? Did it change the way they interacted with one another? Did they become really competitive during particular time periods when resources maybe were scarce, or even kind of counterintuitively when resources were quite dense and predictable? And that can actually cause more territorial behavior because populations can grow. They can begin to stake out a territory that they can defend, and then that could mean fewer interactions between people. So one way we might be able to get at that is through the ornaments that I talked about. And then, of course, how did people eventually lose that hunting and gathering lifeway? Did they kind of vanish and go extinct completely, or did they get pushed up into some marginal areas like those mountains maybe early on? Just to interpret this, don't freak out what you're seeing. What I just want to show you is I pulled this from this recent paper, just showing you that here is the time period we know is represented in Hora, best represented in Hora. So what does Africa actually look like at that time? And what this is meant to show you is that the Central African rainforest was much bigger, and the Sahel itself stretched all the way up to here, meaning that this was all grassland. Very nice grassland for large herds of animals. There were enormous lakes in the middle of the Sahara and rivers that connected them. So this is kind of the green Sahara that you might have heard about. And there's a lot of rock art in the middle there that attests to the fact that people were there, living in very large communities in some cases, fishing along these big lakes, paleo lakes. And of course, now the Sahara goes all the way down to here, right? So if this was happening in the northern part of Africa, there's always the question of, okay, well, what was happening in our part of Africa? And the answer is that we don't actually really know, because there aren't that many data points. A lot of the work that's been done has been very, very regional in scale. So you might get a lake, and you drill down into the lake and you get a great pollen record or something, but then the lake might be hundreds of kilometers away from your study site. Or you might, in this case, this is modern rainfall. You can see that what happens is in December, January, February, the rain is sitting right over our study site. And this is the intertropical convergence zone, which shifts up and down with the seasons, meaning that wherever you have a place that's in the middle, it gets 2 rainy seasons. Whereas Malawi sits at the bottom, so it only gets 1 big, big, big rainy season. And then it shifts away, and it shifts away. So for the rest of the year, here we have June, July, and August. That's why we work in June, July, and August in Malawi. No rain, almost no rain. And then over here in September, October, November, it's the same thing. It's a very, very prolonged dry season. So today you would call it a highly seasonal environment. One of the things we'd like to know is, was it that seasonal in the past? And also, was the so-called African humid period actually dry in Malawi, right? So how extensive across Africa is the African humid period, and to what extent is it possible that maybe it actually doesn't extend this far? You'll notice that most of those reconstructions are focused on the Sahara. Big changes in the Sahara, and we just really don't know what was going on down there. There are some inferences made from very small data sets that things might have been drier or things might have been wetter, but what that does is it sets us up to try to test that. So our new project is going to be to do the archaeology concurrently with the paleo-environmental reconstruction, so that we get the environmental background, and then we also get the cultural response to those environmental changes at the same time. Just showing you an example of how these things work. Here's our site here, and then there's this lovely core from Lake Tanganyika, but that's still almost 500, 600 kilometers away. And that gives us kind of an indication of what's going on 60,000 years ago to today. This is all montane forest pollen, and you can see about 15,000 years ago, it just kind of goes away. There's not much mountainous forest taxa producing pollen that's ending up in these lake catchments after about 15,000 years ago. So this is the period represented by Hora. You can see that it mostly intersects this point when the montane forests are really on the decline. And this is just precipitation. And up here is temperature, so higher is higher temperatures. But I've pulled this from Ivory and Russell paper from last year. So the pollen is where we're really interested because that can give us at least a regional catchment scale sense of what's going on with the vegetation. And we're working with Sarah Ivory on our project. She's actually the person who's studying our pollen, so that's hopefully going to give us some good connections. She worked on the big lake cores that they have from Malawi right next to our study site. They're within 100 kilometers, so that's great. But because they were such long, deep cores, they go back 1.3 million years, they kind of don't really preserve the Holocene. They sort of obliterated the top part when they were trying to get down to the deeper part. So their cores start about here, which is not helpful for Hora. But what we can see is that the last glacial maximum, so the ice ages transitioning here into the Holocene, right? Very cold, very dry, yet somehow more trees, which is kind of counterintuitive. These are the kinds of discordances that we see in the published paleoenvironmental data that come from these huge catchment scale analyses. And what we need to do now is we need to go zero in on the valley itself and get local scale data to try to understand, okay, if I was a hunter-gatherer actually living on this landscape, at the scale that's relevant to me, what was happening in my life, what was happening in my environment at a given time. And to do that, we've got another site here. You can see Hora is this green star, and we're down here, and this is another little shelter. We found it in 2016. So I did not find this one in the literature. It's the first site that I found that Desmond Clark did not find before me. I was very excited about that. And it's got some rock art on it, which is cool. The rock art's up here. It's not very noticeable, but it's present. And then, you know, this is me on the day I found it, trying to record it. So there's all kinds of things sitting on the surface. And the surface doesn't always give you a great indication of what's going to be below it. And it could have been this deep, we didn't know. You know, you can't just go there and scratch around and try to find out. You have to actually plan it, dig it properly, and then you might be disappointed. But we were not disappointed. We went back there in 2017. Here's our excavation. You can see that this person is continuing. He's standing. We got down to about, I would say, 1.7 meters, and it's still going. And we found a human toe, which is nice. Just a toe. That's it for now. But still nice to have some human remains there because it means that there's potential for more. It was a very small excavation. And you can see we're mapping everything with these instruments, right? So everything that comes out of the ground gets a little set of coordinates so we can actually look at these things relative to each other on a millimeter scale. And here is my strat drawing for that. So up here is where we did all of that really delicate plotting and mapping, and then we did this one little half meter where we just went down because time was running out and we needed to do one tiny little test so that we can convince the authorities and the funding authorities as well that we had a site that was going to be productive. And what I'll just point out some of the key findings here. So this is a sondage, a small exploratory excavation. What we found is that the site ranges in time from the present day to 28,000 years ago and ongoing. So this is by far the oldest site in Malawi. There are older sites in Malawi, but not with fossils that are preserved. So this is the oldest fossil fauna in Malawi that's actually associated with archaeological remains. So I keep having all these caveats. There are fossils in Malawi, but no archaeology associated with them. There are artifacts in Malawi that are old, but no fossils. Now here we finally have both in the same place. It goes back to at least 28,000 years ago, and it's got these ostriches again. There they are. So they're all in there. There's more ostrich fragments here. And you can see that our dates are generally good, but we have this funny one at the top that's very old. And I'm interpreting that. We know over here there's a pit that somebody dug in the last 200 years. And I think that they've pulled up some old material and dumped it on the top. But it's a little bit of an ad hoc or post hoc interpretation, but we're still at kind of the exploratory phase of this site, and we can test that. And then, of course, down here, there's a modern piece of charcoal. That was annoying. And I hadn't dated any of this other stuff, and I thought, oh, great. But then we went and we got some snails, and we dated the snails, and we dated some other charcoal. And we have this beautiful sequence, right? 5,000, 9,000, 23,000, 25,000, 27, 28. So it's all coherent except for this thing. And imagine, I mean, you saw that guy. He's in a little, he's a very thin man, but he was in a pit that was this big, right? And he had to excavate by bending over straight over. So it was when he would kind of like unroll when he would come out of the pit. And there's a lot of potential there for things to sort of fall in. So I'm hoping that explains why, because there shouldn't be charcoal down there anyway. There's no burning features that we can see. And he would have seen, because they were instructed, when you see charcoal, grab it. So he would have been like, yes, charcoal, and grabbed it. But look, I mean, we'll go back and we'll get a bigger excavation and we'll test that. So here I am again here, and I just want to fill in this gap here with Mazinga. So this is the other site. Much longer coverage. And it goes all the way back into when we know that this montane grassland seems to have been, at least on a very broad regional scale, indicating cold, dry, yet somehow more wooded conditions. But we're just gonna have to see. We're gonna have to see what the local environment tells us. And to do that, we've got this lovely section, which this river is right outside our site. The site is here, and this big section is here, and it's about 3.5 meters of just layers and layers and layers and layers of old soils, old pollen, old stuff that has been building. We don't know how old it is. We're in the middle of trying to get our results back, but I hope it's very old. and if it is, then we will at least have a local paleoenvironmental sequence that we can match up to those and contrast with those fine resolution but far away lake cores. So that is the hope. There's a pollen grain there. And lots of charcoal. So there's this whole area is just full of, it records old burning events, old soil formation events, which indicate changes in the environment. There's either more precipitation, more organic activity. Lots of things can cause soils to form. And you've got a series of them here. So that's cool. And then here are the ostriches again. So they're back. We found several unmodified ostrich eggshells. Some of them are, when I say unmodified, I mean not made into beads, but some of them actually have a little bit of, like this one has a little bit of red coloring on it. This one has a little scratch on it that looks like an incision, and it is known that ethnographically people will decorate their ostrich eggshells, and they will use them as containers, like water containers to drink out of. So finding these was very interesting to us, because typically if you find the fragments and not the beads, it means that the whole egg is moving, as opposed to just beads or ornaments that somebody might be wearing on their neck. And hopefully what we can do is get some information about the environment from these guys. So you can see how there's a modern Khoisan person, and he's using one as a container. And Desmond Clark, again, said that there was an ostrich at this site, Zawi. So our site's up here, Zawi's down here. You can see that's about 400 kilometers away, and it looks like that. I don't know. I mean, you can maybe write down what you think it is and anonymously pass it up at the end of the lecture. But that's what he's saying is an ostrich. And this is evidence that the ostriches used to be there at the time when people were recording it. And I think it's ambiguous. But this is what I was talking about. Modern ostriches live up here, right? And they live down here. And they don't live in here. This is Malawi right there. And it's like the whole of Malawi is outside the ostrich zone, not just our little piece of it. They don't live in this entire Zambesian phytogeographic zone. It's just not great for them. It tends to be characterized by lots of low, scrubby brush, and they like open grasslands. So we have these kind of subspecies of ostrich, Masiacus and Australis, and the genetic data from living ostriches shows that in the recent past, these two were exchanging genes. So there is some biogeographic data that suggests that there should have been some kind of ostrich exchange happening in the middle here. And we're probably picking that up with our eggshell. So the question is, what was the environment like at the time? And we've tested the eggshell for oxygen isotope ratios, which can tell us about changing precipitation regimes. And then you compare those ratios to other known parts of Africa where you have known values of these ratios of oxygen isotopes. And some of them compare to up here, right here in the very arid Omo. So it could have been that arid down here at the time that we're picking up some, that people were clearly living in the area. They're living there because they're bringing the ostriches in. So that's an interesting result. And we're really keen now to try to get some ancient DNA from those ostriches. So maybe that will help us resolve the problem of, the biogeography of this species. Right, recent connection. And one other thing we're going to do is this, what you're seeing here is our 2 sites. These are those things that we sampled. These boxes are those profiles. We have several profiles with old paleosols. So pollen, we're going to date them. We're going to find out what the local precipitation was like, hopefully, and the local vegetation. And then what you're seeing here in the color gradation is a change in the ratio of strontium-87 to strontium-86. And that's dictated by bedrock, amongst other things. So it's a naturally occurring ratio that just varies across the landscape. But anything that lives on that landscape will incorporate that same ratio into their bones and hard tissues as they're growing up, which means that if we find an ostrich eggshell and it has a strontium ratio up here, and we find it down here, then we know that that ostrich egg has moved at least that far. And we're going to do that with the ostrich eggshell. We're going to do it with the beads, the snail beads, and also with the ostrich beads. And that will tell us if it's local or non-local. People are just going out and getting it locally, or if they're actually trading these things very extensively around the area. And this is going to be one of our proxies for trade. And then we're going to look at the tooth enamel of the fossils that we find in the site, look at their ratios. And if we find that there's a huge amount of ratio around this site, for example, that'll tell us that hunting ranges are really quite big. And if we find a very small number of ratios in terms of the diversity of ratios, then we'll know that the hunting ranges were small, and that would indicate more territorial behavior on the part of hunter-gatherers. There's kind of a series of inferences you have to follow. But having access to the strontium isoscape is incredibly interesting. And we've just gotten permission to sample the human teeth from Hora, so we'll know where they came from, too. And what's very cool about it is the teeth that are growing when you're younger, they will take on the strontium ratio, isotope ratio of where you develop those teeth, so your childhood. And then, so your M1s, for example, when they're growing, will have your childhood location sort of embedded in them. And then as an adult, you will have ratios of where you spent your adult life. So if somebody has moved over the course of their lifespan, and you can get a young developing tooth and an old developing tooth, you can actually see that lifespan movement. And we might be able to even begin to look at exogamy. I mean, it's a sample of 2, but it's still going to be very interesting, I'm sure. Right. So just to kind of close off that question, what was life like for hunter-gatherers? That's one of the things from my abstract. We made some predictions. We predict that during arid periods, human groups should actually expand their hunting network, that they should interact more with one another. And we know that this is a risk reduction strategy that modern hunter-gatherers use in places like the Kalahari. So they reinforce these relationships through kin exchange, usually wife exchange, and then also the exchange of goods and materials, ostrich eggshell containers being amongst them. Ostrich eggshell beads also being very important. So we should see a larger diversity of these strontium ratios at times when the environment was arid. During times when it was wetter, we would expect those hunting ranges to contract and people to start focusing more on local resources, especially stuff you can get out of rivers like fish and crabs and things. And we find some of that in the sites. So far, our sample is not enormous, but it's certainly suggestive that there was some change going on. And then there's the question of the people themselves. So this is what Kendra is mostly going to talk about. And I have to put this up again. This is Clark's own words, and this is the language that people used to use back in 1956 to describe different populations of people. It was all about classifying people. So do they belong in this type, this type, this type, or this type, right? And of course, a lot of these words are not words that we would use anymore today, but that was the obsession back then, was if you find a skeleton, who does it belong to? And they would do various measurements on the bones to try to figure that out. So Clark was saying, hey, these guys are probably just a mixture of everything. This was his prediction. Now remember, he predicted how old these skeletons are going to be, and nobody really thought that that could be the case. And then we actually found that that was the case. So we'll see if he's right about this one as well, based on his limited knowledge. There was a reanalysis of these skeletons done in 2006, where they say that they reject the hypothesis that they were just Bushmen that happened to live in Malawi, right? So your Khoisan, sometimes they prefer to actually be called Bushmen themselves. The idea being that, oh, everybody who used to live across Africa was a hunter-gatherer. They were all just Khoisan people. And they're saying, no, they don't really look like Khoisan people do skeletally. And they couldn't really peg, again, which population they looked most similar to. So they kind of look a little bit like a lot of different things skeletally. So we'll see what the DNA tells us about that one. And I suspect I'm probably running out of time.
There's, well, 25 minutes total left.
Yeah, okay. I want to make sure you've got your time. So what I'll do is I'm just going to take you super, super fast through the next thing, which is what happened to them. Right? So you just hang in suspense about who they were. And now we're going to look at this question. Did they vanish completely? So what you're seeing here is these are modern DNA samples from Tishkoff et al. She's worked a lot in Africa. And what she's done is she's taken a bunch of samples of different people who are considered to be indigenous to a particular part of of Africa. And what she's found is that you can see that the recent or current hunter-gatherers usually look quite different in terms of the proportions of haplogroups that occur in a population. They look different than the people who tend to surround them. So it's not that they don't admix with folks around them, it's just that they don't admix as much. And they seem to have had a different genetic history than the people who were living there as the majority group today. Same thing down here with your Khoisan people, right? So you can see that there are people with this light green. The light green dominates in Khoisan ancestry, and it's just kind of there in these other populations. So there's some admixture, but they're clearly different in terms of the proportions of genetic composition that they have. So that's interesting. And when you also look at the language groups in Africa, I just want to point out that these turquoise-colored ones are languages that include click consonants. I won't try it, but the kind of clicking sounds, right? Thank you. So they exist in two parts of Africa, and they aren't connected to each other. It's kind of reminiscent of the ostriches, isn't it, right? Because like this whole area is blank, and then you have them up here and up here. So there's kind of this sense that maybe that's the ancestral language, and that ancestral language group used to be across here. So what's up with this big blue one then? That seems to be the result of what we call the Bantu expansion. So sometime around 4,000 years ago, there was a group of people who lived in this region, and they developed ironworking technology, agriculture, pastoralism, ceramics, not necessarily all at once or all at the same time, but all of those things are associated with a group of people who ultimately expanded either in one migration or many out of that region and basically just took over the rest of sub-Saharan Africa. So they brought their languages with them, and they also brought all that stuff with them too. So archaeologically, they leave kind of a breadcrumb trail that you can follow. And genetically, so this is an archaeological phylogeny. It looks really complicated, but all it is it's showing you, if you look at that southern part of Africa, cultural materials like pottery styles that are more similar to each other tend to group closer to each other geographically. And that makes sense because you're going to share more sort of cultural information or knowledge with someone who lives closer to you than somebody who lives super far away. So what you're actually probably seeing is the result of an expansion from this area and then a diversification as people are moving out of that area and leaving behind different styles of ornaments, pottery, and metal. So I'm not going to have time to discuss what's potentially problematic about that, so I'll just tell you. It's, obviously there can be convergence, right? People can come upon the same solution or the same stylistic preference completely by accident. They don't necessarily have to have a shared common ancestry, but it's a common problem with any phylogenetic approach where you're trying to reconstruct ancestry. You're always going to have the problem that you might just end up with the same result, not because there's a shared common ancestor, but because of just convergence. And that could be the case in some of these cultural things. But the overall pattern is very clear, that most of the diversity is here early on, and then it tends to kind of spread out. This is a really complicated map where I went through so much literature to try to make this one slide. Not for this talk, don't worry, for my other class. But I just couldn't get my head around all the different arguments and pieces of evidence. And what I've done here is I've just made a bunch of circles that show you the main elements of this. So large stock, this is cattle. Small stock, sheep and goats. Ceramics, right? First plants that are domesticated, and then ironworking. So you can just kind of follow this. They probably have an independent origin up here and down here. And then this one expanded this way and then that way. And then some of those elements made it down the east coast as well, but not all. So you'll see here that pottery and sheep came down the east coast, but not metalworking, and also not cows. And then all of that went this way. So Malawi being right in here, it was kind of at the tail end of this enormous population event. And we've picked that up in our final site, which is this cave here called Katawanda. And you can see here the dates, right? They're right in that time zone. You've got about 1200 to 1300. We don't have a date from here. I'd like to know what that is. But down here we have hunter-gatherers. So hunter-gatherers transitioning to that Iron Age group, basically a group of people coming through, bringing things in like these cowrie shells, which they're trading in all the way over from the Indian Ocean, which is very far distance from here. And then coming and digging pits. Here's a pit. Somebody has made a pit here, and then we dated it to about 1,000 years ago. So it's a site that's up in the mountains, and our prediction would be that hunter-gatherers might have been pushed out of that nice fertile valley up into the mountains, and they might persist a little bit longer as hunter-gatherers up in the mountain area. So this is the site we're going to use to test that hypothesis. All the other sites you've noticed are too old to really have that much bearing on that Bantu expansion event. But this site will be able to tell us something about it. And this is just showing you how you can take some pieces of that sediment and you can cut them up and look at them in thin section and actually see micro events like little tiny plant pieces and sediments and charcoals and things, and how they're organized inside the sediment. So to answer that question, I do think that we're going to see some evidence of that mountain area here being used as a refuge, and that we're going to see a lot of variation in hunter-gatherer lifeways over periods at least spanning 28,000 years in that valley. So this is going to be the next focus of research. And then just some pretty pictures of the rock art in the area. It's always lovely to take students to see the rock art. And there's more rock art there. And this site is one of the ones Kendra's going to talk about because there are human remains here. So here and here, ancient DNA. And just to show you, that's what the rock art looks like up there. Okay, so that is the archaeological part of it. And then next you're going to hear about the DNA. And I apologize for the length. I thought it was going to be shorter, but it wasn't. And I cut it short too. I had so many extra things. I just cut. So sorry, I love this stuff. There's so much to say. But one of the things that I think is interesting is that somehow, even in the almost complete absence of any reference points, Desmond Clark got the dates right and seems to have gotten some of the cultural ideas right as well with respect to differences in rock art style. And now we're going to see if he got the genetic part of it right too. So these are my acknowledgments. Thank you so much for listening to me. I'm going to pass it over to Kendra.
Okay, we'll get Kendra set up with the mic around her head. I think some of us are going to have to audit a course or two, Jessica, to fill in the rest of this.
Yeah, that's a lot. Okay. Yeah, okay, that's all right.
So we've got 20 minutes total till 1 o'clock.
I'll be brief. All right. Okay.
You were seeing the end of the flyers that we hand out to people around us. So we always get curious people coming, what are you doing? What are you looking for? And they often think that we're looking for something very valuable, like diamonds, right? That happens.
Blood diamonds, right? Here you go.
So we've made these educational flyers in 2 languages that we can kind of pass out to try to help, I guess, spread the word.
Okay. Hi. As Gretchen introduced me, I'm Kendra. I'm a student of Jess's currently, and I'm hopefully finishing up in the Department of Anthropology this semester, pending Jess's approval of my dissertation. So if anyone wants to put in a nice word on the way out, you're more than welcome. The last time I gave a talk that was very similar to this was in front of 1,004th through 8th graders in Ireland for Irish Science Week, and someone chucked an paper airplane at me that said, "You are very boring." So if you want to do that, please refrain. I'm still getting over first. Okay. As Dr. Thompson said, I worked specifically with the ancient DNA for this Malawian project. And unlike her talk, which was such a beautiful and broad introduction to history and archaeology in Malawi, I'm going to focus much more narrowly on ancient DNA. So you have to have seen evidence of ancient DNA research in the news. It's all over the place in all different sorts of media outlets, right? So we see a lot of this. There's some, I just keep adding. I keep adding as I do the talks like this more and more, and so I just keep on going on and on. But this is the one that you'll notice that we talked about, and you'll actually see Jess's picture of Hora right there. And this was, I think, Smithsonian. And I just put this one on because this was even a more recent article, beginning of January, helping to using ancient DNA to look at the settlement of humans into the Americas. A single genome was published from a young girl up in, I believe it was in Alaska. So ancient DNA in the news, it's very popular today. It's very sexy. People love it, and it's interesting because it's telling us this story about ourselves that we didn't know and we didn't have the ability to reveal until very recently. So what is ancient DNA, before we begin? Ancient DNA is the study of DNA that's recovered from biological specimens that are anywhere from hundreds of years old to hundreds of thousands of years old. So right now we believe that the limit to DNA preservation is probably about 1 million years. The oldest study for humans is about 450,000 years old, and that's from a cave in Spain, actually. But so this is normally what you think of when we say we're analyzing human DNA. But the actual specimens we analyze aren't always human. In fact, there's been a lot of really brilliant ancient DNA studies done looking at cattle, dogs, cave bears, and even penguins. So this is looking at things like domestication events, more so of cattle than of cave bear, obviously, but also then looking at things like evolution, like cave bear genomes in comparison to modern bear genomes, and how these genomic changes are taking place over large time scales. So it's been very informative. There's actually even been some really interesting studies lately on studying ancient DNA in maize. So I focus on human remains, so that will be the focus for the rest of our talk. So what we want to know is, how do we go from a specimen that looks like this to data that look like these? And these, you know, while Jess might more comfortably speak this language, this is the language that I'm more comfortable working in. So that is a little bit more catchy. How do we go from bones to bytes? All right, so from skeletal data down to genetic data. So we face a lot of challenges while we try to do that. The first and most limiting factor is degradation. And this is because DNA degrades over time. It is a biomolecule, and it is naturally degrading once the enzymatic processes that repair it cease to function upon an organism's death. So it makes sense. So we have trouble producing sufficient quantities of ancient DNA. And often the times the quantities of DNA that we do recover are highly damaged. So we see things like hydrolytic damage from obviously from water, but that results in cytosine deamination. So this is the loss of an amine group at the ends of the molecules. We also have a lot of issues with contamination. So this comes from environmental DNA as well as modern human DNA, which PCR would much rather amplify my nice, complete, long, undamaged human DNA than these little modern fragments. We also have issues with organic PCR inhibitors, which can be things like tetracycline. And anyone who might have known George Armelagos would have known that he studied tetracycline in the Nubian skeletal remains, which turns out to inhibit PCA and have downstream effects on your endogenous ancient DNA yield. So these are kind of interesting things to consider. So in order to do ancient DNA, which is what we want to talk about, there's steps of ancient DNA analysis. And the first is to sample the skeleton. Okay, so what we know is that dense cortical bone preserves ancient DNA much better than spongy trabecular bone. And this is probably largely just due to bone density. All right, so here we have a long bone, and you can see the cortical bone. If we had this bone, and that was our only sample, such as a lot of the specimens at Hora, we'd want to sample from here as opposed to here. Second, we found out that the petrous part of the temporal bone, so here you see the temporal bone, this is the ectocranial view, and an endocranial view, with the petrous portion highlighted in green. So that's this little pyramid right here, and here's a picture in real life. We found that the petrous bone preserves ancient DNA better than any other bone in the human body. And this was an empirical test to show that. So from each of these specimens, you can see here there was a metacarpal and a piece of petrous, and this is the endogenous percent of endogenous DNA that was recovered. Okay, so in all the cases, the petrous bone gave the best DNA recovery. And why that is, we're not really sure. There is a definite possibility that it has something to do with the embryonic development of this part of the temporal bone, which is known to ossify very early in utero and undergo a reduction in remodeling throughout life. But we're also trying to find out right now specifically where DNA is located in bone. And so until we know that, this is largely a mystery. Building upon this finding, and this is a large part of my research during my PhD, is we took one petrous and we divided it into 3 different areas. This A area is trabecular bone at the apex of the petrous. This B area is cortical bone, which surrounds the C area, which is your osseous labyrinth. So your osseous labyrinth is your cochlea, your semicircular canals, responsible for hearing and for balance. And we conducted an intra-petrous comparison, taking a bunch of different bones and comparing these 3 different areas. So what we found is that part C, which is the osseous inner ear, preserves DNA better than the cochlear portion by up to 65-fold, and from the trabecular portion of the apex by up to 177-fold. So now we know this is the area that we want to target. But there was an additional question here. Okay, that's right. Okay, so what we want to do is target this piece of bone through the grinding lab. So we take our petrous, you can see right here, and this is how we get our bone sample for DNA analysis. And we take our petrous, we thoroughly clean that petrous in an ancient DNA clean room, usually using the sandblaster. Same sandblaster you can order on Amazon. Really, that's where we order it from. We isolate the area of the bone that we want to study, which is this osseous inner ear. Here you can see the cochlea, and here are the semicircular canals. And we grind it to a homogeneous bone powder. Okay, and this is the substrate that we use to extract DNA from for the analysis. But what happens when the skull is intact? How do we get to that intracranial piece of petrous bone without destroying a whole skull? So here we go. Here is the part we're targeting, actually inside. And what we do is we actually use a dental drill, a Dremel drill, also available on Amazon. I'm not paid by Amazon, believe it or not. I'm sure at least somebody has a Dremel drill in their, yeah, as I say, someone's got to have one in their tool shed right now. Don't get any ideas. So what we do is we use this Dremel drill to approach the skull from the base of the cranium, which is actually a method that I developed and published last year to directly access that DNA-dense osseous labyrinth of the petrous bone while causing minimal damage. And so this is important for the future of archaeologists and osteologists and geneticists working together while we care to preserve their human remains and they care to make sure that we get the data we need for genetic analysis. So this is a good step forward in making sure that this relationship stays productive and cohesive and positive. So our next step, once we have our DNA powder, is to extract DNA from it. And the way that we do this, I'm going to skip a little bit of this for time. But the way that we do this is basically to immerse this bone powder in a buffer solution that will be able to release the DNA from the bone molecules, right? And here's a picture of me. It's one of the only selfies I've ever taken working in the ancient DNA lab. And the reason I include this is because this is the equipment that we wear to actually work in an ancient DNA lab. And this is because it is a sterile environment, and my DNA wants nothing more than to contaminate these samples. So this is the way that we try to prevent it and protect as much as possible from contamination. Our next step is library preparation, and this is nothing to do with books. It's about preparing DNA sequencing libraries, which are taking the DNA fragments that we extract, fixing them, making them nice and blunt at the ends, much more organized, adapting these, filling in these little adapters to these molecules to make them visible to sequencing technology, and finally, attaching an index or a barcode. So this is a 7-figure nucleotide sequence that we just tack on. Okay, you can see, there we go, animation, tacking it on right to the end here of our molecules. And what these sequences do is enable us to pool multiple samples together for sequencing, and at the end, separate them out into each individual, if that makes sense. So that means on one sequencing run, we can put on 10 samples, and then at the end of that sequencing run, we could easily separate the DNA sequences that belong to each individual, and that's why we do that. Our next step is target enrichment and capture, and this is where we take a mishmash of DNA that is contaminant and human, and we capture it actually using synthesized bait sequences that are complementary to the human sequences that we want to analyze. By combining our bait library with our sequencing library, we're able to hybridize the bait molecules to the human molecules and wash away all the contaminant and capture only the human molecules. Okay, so that's the way that we take highly contaminated DNA libraries and only sequence the human DNA. So that makes it more economically efficient, that makes it more time effective. I'm not analyzing 99% of data that I will end up throwing out, right? It's just human. And finally, we sequence this capture product, and I'm actually going to fully skip this because it's a bit technically complex, but we get all the genome sequences we need and we analyze them. So our last step, and this is what I want to talk about a little bit in relation to the Malawi project, is our DNA data analysis. So the first thing that we'll do is we'll look to quantify the damage pattern from the DNA we extracted to make sure that it's actually ancient. And what we expect to see is this pattern right here, where the substitutions at the end of the molecule are increased in relation to the rest of the molecule. So this means the end of the molecules are damaged. The wrong base has been incorporated at the end of the molecules because of these strand breaks. So we see about a 50% frequency in damage at the last base pair and maybe 12% over here at the other end of the molecule. So this tells me that the DNA I'm looking at is ancient, where something like this tells me that we don't really know, and that's probably a lot of contamination. So we're looking for a pattern like this. Second, we're looking to determine the sex of the individual. So what we're actually looking for here is to take the number of reads and plot this against the length of the chromosome, recognizing that longer chromosomes receive more reads than shorter chromosomes. And fortunately, someone decided to number our chromosomes from longest to shortest. So by this beautiful linear scale here, we can determine, based on the number of reads on the sex chromosomes, if an individual has one X or two X's. So if they have two X's, they'll fit right along this linear line, which is diploid. So basically, we'll have one copy, or we'll have two copies of chromosome 1, 2 of chromosome 2, etc. So if we have 2 of chromosome X, we expect it to fall right on this linear pattern. If we only have 1, we expect it to fall about halfway down, which you can see. And this method is still unpublished, though we're quite confident in the conclusions we can make from it. We also assign mtDNA haplogroup, which Jess alluded to very quickly, knowing that mutations on the mitochondrial genome accumulate sequentially, and they can be used to trace radiating maternal lineages around the world. Okay, so here's our tree. We know this tree looks like, we know what this looks like, and all of these different haplogroups are primarily found in specific geographic areas, which you can see from this map here. And a lot of these, Africa specifically is very unique in its distribution of mitochondrial haplogroups. And this spreads throughout the rest of the world, but you can see very clear patterns here. So that's an important part of helping to assign matrilineal ancestry to our ancient samples. The other thing that we do is to look at ancestry relatedness, and this is one of the important things that Jess alluded to in her talk, how we're going to see which modern day populations might share genetic affinity with our ancient individuals. So here you can kind of see, this isn't a fantastic chart, but there's a modern day backgrounds where an ancient DNA samples are plotted onto that modern day background. So this is a bit confusing because it's in color. So we'll talk about that a little bit more when we get to the Hora material. We also look at things like ancestry components. So we take a more subtle and nuanced look at geographic affinities, and we look at ancestry, and we look to divide the genome into different components of ancestry and model that in comparison to a known genetic landscape. So we might take present-day individuals from a bunch of populations, tack on our ancient individuals, and see who these look like and what kind of proportions of ancestry are being shared between groups. Okay, so we approach the ancient DNA analysis of the Malawians in this paper, recognizing that the genetic population structure of Africa prior to that Bantu expansion that brought genes, agriculture, ironworking was largely unclear. So what we wanted to do is reconstruct African population structure prior to the spread of food producers. And the reason I highlighted this spread of food production here is it's not food production in itself we cared about, it was the genetic consequences of having people who brought food production move into new areas, basically. So it's a little bit more complicated than that. So as I said, we analyzed 7 ancient Malawians as part of a larger set of 16 sub-Saharan Africans, and this did increase our sample size of sub-Saharan African ancient DNA from 1. This was huge. This was a very big study. And the Malawians we analyzed as part of this ranged from 8100 BP to 2500 BP. So we had a very large time scale as well. So we confirmed our post-mortem degradation patterns. That was that nice map damage plot I showed you with increased damage at the ends. We determined sex, mitochondrial haplogroup, and Y chromosome if the individual is male. And we looked to understand how genetic diversity changed throughout time in Malawi and used principal component analysis to relate ancient individuals to present-day East Central Africans. So what we found here is a very clear climb of ancient hunter-gatherers that ranged from South African ancestry over to East. And the Malawians fell right in the middle, as you would expect them to geographically. Okay, so you can kind of see the climb right here, as well as the climb right here. And this guy, a Tanzanian hunter-gatherer, is a huge outlier, even though he kind of does fall within this climb. But if you look at his date, he's 600 BP, as opposed to the majority of individuals who are much older, so I'm not surprised to see him as an outlier. Another interesting thing we found is that here are our Malawians, right here with this coral, yellow, and black component. These are our Malawian hunter-gatherers. They're all pretty genetically homogeneous, and this is across a period of 5,000 years. So we see a long-term stability in genetic structure over this time. However, right here are modern-day Malawians, and what you can tell is that they are not at all similar to the people who lived in this area before them. And that was interesting in itself. So what we were able to determine that in between roughly 2500 BP and the present, there was a complete population replacement by Western African food producers. So basically, most likely in association with the Bantu expansion as well as other smaller scale demographic events that occurred following that time. But, okay, no problem. I think that is my last slide. Okay, so implications. We've expanded our data set a lot. We highlighted the power of ancient genomes to provide insight into prehistoric events that you simply cannot see with modern data alone. And we revealed this presence of this hunter-gatherer climb that was not known before and will be the subject of much further study. And that's it. Okay, thank you.